Good afternoon, honors biology students. We tried to do this earlier this morning, and for some reason it wasn't working, so I'm trying this again. So hopefully this will work. This is the second time I've done this. Today we're going to talk about human genetic disorders, and specifically we're going to look at abnormal chromosome numbers in the human genome. In order to do this, what we're hoping to do is explain how that happens through the process of meiosis, hoping that maybe you do remember something about how meiosis occurs, the making of gametes, and then describing how small changes in DNA can affect human traits, which we pretty much already talked about in previous lectures. The main thing we want to talk about today, and I'm actually going to send a video along with this describing exactly, specifically, how this works, non-disjunction. We've mentioned it before. We've talked about a little bit about it in might have been back in chapter 13, chapter 12, how this can cause chromosome number problems. Non-disjunction is when homologous chromosomes do not separate as they should during the process of meiosis. And I guess we should probably go back and do some review on some terms. Homologous, the word homologous refers to same chromosomes, which means in the human cell, in a normal body cell, or a diploid cell, we're going to have 23 pairs of chromosomes. And those chromosomes are going to carry the same information. They'll be carrying traits for the same, or genes for the same traits. So that's why they're called homologous chromosomes. So we have 23 pairs of those, and in each one of those pairs, they're going to be carrying the same traits. Now, they may have different alleles for those traits, but they're carrying the same traits. So that's why they're called homologous. Now, during the process of meiosis, we're going to be creating haploid cells, which are called sex cells, or gametes. In this process, there are two divisions, and the problem comes about when there's not separation in meiosis 1, during anaphase of meiosis 1, there's not separation of the tetrad. If you remember, the tetrad formation is when a pair of chromosomes duplicate during the meiotic process. When the meiotic process starts, they duplicate. So up here you can see on this diagram where each of these red chromosomes and each of these orange chromosomes have made a copy of themselves. So now we have 4 chromosomes, and during prophase they will remain together. Now they don't show them stuck together here, but they form what's called a tetrad. During anaphase of meiosis 1, the first division, these 2 pairs are supposed to separate and go to opposite cells. When they do not do that, that's what's called non-disjunction, and typically this happens during the first division of meiosis, so in meiosis 1 as it's called. So what happens is when these two do not separate, like let's say in this particular case, the red chromosomes have separated, so we have a pair of chromatids here and a pair of chromatids here for the same traits, but the tetrad of the yellow did not separate. All 4 chromosomes went to one cell, and the other cell had none. So the result on the second division is that these cells will have one of each of the one member of each of the chromosome pairs, except for the yellow chromosomes, which it will have none. So if these cells, if these gametes are used in fertilization, that individual will be missing a chromosome. It'll be missing in this case, the yellow chromosome. Now, we'll have a yellow chromosome from the other sex cell, but that will only leave it with one member from the original pair. Over here you can see we also have a problem because this one, which did not separate in meiosis 1, is now going to produce 2 cells that are going to have an extra chromosome. They should have one of each of the original pair inside the cell, inside the gamete. As you can tell, these 2 have an extra yellow chromosome. So non-disjunction has occurred there. Keeping in mind that we have 23 pairs of chromosomes, 46 chromosomes in a normal body cell. In a haploid cell or a sex cell, a gamete, we're going to have 23 chromosomes, 1 member from each original pair. What determines a male and a female is whenever fertilization occurs, whether there's the presence or the absence of a Y chromosome. So the chromosome pair number 23, which is the sex chromosomes, if you have an X and an X, that typically designates a normal female. If you have an X and a Y, that designates a normal male. We can have extra sex chromosomes or missing sex chromosomes due to non-disjunction, as well as the autosomal chromosomes, the other 22 pairs. We can have missing chromosomes there, or we can have extra chromosomes there due to non-disjunction occurring in meiosis 1. Now, the question usually arises, can this happen in the second division? And the answer to that is yes. If this happens again in the second division, that means that one cell is going to get the whole complement of chromosomes and the other is not. So it can happen in meiosis 1 or it can happen in meiosis 2. Typically it happens in meiosis 1. And I will say also, this type of a mistake that occurs typically occurs more often in the egg cell, and it more often occurs in folks that are of reproductive age but are a little bit older in reproductive age. It can happen in the sperm cell. It doesn't happen as often in the sperm cell as it does statistically in the egg cell, but it can happen in both. Now, one of the things that people do when they're having a baby, when they find out they're pregnant, is a lot of times they'll do something called an amniocentesis. And an amniocentesis is basically taking cells that is sloughed off from the fetus inside the mother's womb and sticking a needle into the womb and taking out some amniotic fluid, which is the fluid that surrounds the baby. And this fluid will have sloughed off cells from the baby, and they analyze these cells and take them through karyotypes to see if there are any abnormalities, some missing chromosomes or extra chromosomes. That leads us to actually an ethical problem where in some cases people may choose to terminate the pregnancy if there is some abnormality there. But amniocentesis can absolutely be a test that will tell the parents whether their chromosome complement of their baby is correct. So let's talk about the most common of these abnormalities. The most common abnormality of chromosomes is something we call a trisomy, and it happens in trisomy in the chromosome number 21. So we have trisomy 21, and that's called Down's syndrome. Trisomy meaning there are 3 chromosomes in the number 21 pair instead of 2. Now, we've already talked about Down syndrome a little bit in class before we left, but you also need to know that Down syndrome, the same characteristics that show in the Down syndrome person can be manifested by a 15-21 translocation. Let me refresh your memory on a 15-21 translocation. That means part of the 15 chromosome in a specific area likes to break off, and it likes to reattach to the 21 chromosome. So one of the 21 chromosomes has more genes in it than it should. It causes the exact same symptoms as a trisomy 21. They both show Down syndrome characteristics. Now, If you are a trisomy 21, you are a Down syndrome individual. Some of the characteristics that you'll show, you will have 3 number 21 chromosomes, and this happens in 1 out of every 31,000 births. So it's not something that happens a great deal of time, but it does happen. These individuals are generally short in stature, have short, broad hands, stubby fingers, small, round face, protruding tongue, protruding thick tongue, sometimes a webbed-like neck, mild to severe retardation, mental retardation, an increase susceptible to other birth defects and diseases. These people were sometimes, some time ago, called Mongoloid, and they were called Mongoloid because of their Eastern look, because they had narrow slanted eyes that were an Asian type of characteristic, and so they were called Mongoloid. The politically correct term now is obviously Down syndrome, but those eye characteristics are a main feature. And these individuals are sterile. So an extra 21 chromosome is absolutely not a benefit. You have to, if you have 2 21 chromosomes, you're all right. If you have extra, that doesn't mean it's better. That means it causes problems. So that's one of the most common of the chromosome characteristics that can go wrong. Non-disjunction of the X chromosomes can lead to a disorder also known as Turner syndrome. Turner syndrome is a situation that happens quite often in females. Statistically, 1 in every 5,000 females is a Turner's female. And in this particular case, a Turner's female will have 1 X chromosome missing because of non-disjunction. So we call these individuals the XO female, or a 45X, meaning they have 45 chromosomes and they have 1 X chromosome in their sex chromosome complement. If we were talking about the Down syndrome person, we would say that individual is a 47 X or XY because it has an extra number 21 chromosome. Now, the Turner's female, a lot of those individuals are not going to make it to birth. A great number of those undergo spontaneous abortion, which means basically that they are not developing correctly, and at some point they die and the baby is aborted naturally by the mother. Those individuals that do survive are generally sterile because they lack sexual development. They don't have the extra X chromosome. They lack normal sex organ development. They are within the normal range of intelligence, and they are extremely short in stature. When I say extremely short, they're well below 5 feet. They're in the 4-foot range. So they have a number of very distinctive characteristics. Another of the sex chromosome characteristics that come up from non-disjunction, a very common one, would be Klinefelter syndrome. This is a 47XXY male, which means these males have an extra X chromosome. They have the Y chromosome, so that indicates that they are a male. Now, these individuals will be sterile, and they have some more feminine type characteristics. They have scarce beard, very delicate skin. They are pretty much normal looking males, though. They have usually low mental ability, not in the mental retardation category, but low mental ability. And These individuals, this is one of the most common of the male sex chromosome abnormalities due to non-disjunction. Now, keep in mind there have been no reported cases of babies that are born without an X chromosome. So if you have an you have a Y chromosome, which indicates maleness, and no X chromosome, those individuals do not survive, keeping in mind that the X chromosome carries a whole bunch of normal characteristics that a person needs to survive. So if a person doesn't have an X chromosome, they are not going to survive pregnancy. They will be spontaneously aborted. The Y chromosome, if you can remember from when we talked about that, contain a sex-determining region on them that is necessary for normal male development. Even if there is several X chromosomes, as long as they have the Y chromosome, the male will survive and it will be a male. There's also something that was talked about years and years ago as an antisocial behavior type characteristic, and that came about, that idea came about from individuals that were XYY, individuals that had an extra Y chromosome. And it was studied in prisons because of a famous serial killer in Chicago back in the 1960s, a man named Richard Speck. He was convicted, and when they did a chromosome complement on him, they found he was an XYY person, and they started doing testing of a lot of prisoners in prison to see if there was maybe some connection between an antisocial behavior having an extra aggressive complement to their behavior because of the Y chromosome. Thorough testing over the years, though, has kind of put some doubt onto that category, because there are males that are XYY that have lived normal lives and have not displayed that antisocial behavior. So that is just another one of the fairly common of the male sex chromosome disorders due to non-disjunction. One of the things that we've also talked about before is when we talked about protein synthesis, we talked about the idea that there has to be the exact sequence of nucleotides so that we get the exact sequence of amino acids in every protein that is coded for in a gene. So changes in the DNA sequence of a gene can change those proteins. It can alter the amino acid sequence, and it can affect the phenotype. In this particular case, what we're looking at here is one of those altered proteins. The protein is called hemoglobin. And in the case of hemoglobin, where we have a substituted amino acid in a wrong position, and it's causing cells, as you can see, instead of being biconcaved, oval, rounded type cells, to turn into these sickle-shaped red blood cells that tend to clog up circulation and cause problems and don't carry oxygen very well. So that's all due to one amino acid being out of place. Another of the proteins that is not being made correctly because of a mutation is the CFTR gene, which basically produces normal mucus, and it makes a person produce normal mucus. If you're missing that particular gene, or you're missing the three bases in the DNA that code for a particular amino acid, that means that amino acid has been deleted and a person does not produce normal CFTR, which is basically mucus. And that person then will produce very thick mucus, which will clog up a person's lungs, clog up a person's digestive system. Anywhere that there is mucus being produced on a regular basis, the mucus is going to be produced there is going to be very thick. So That causes cystic fibrosis, and it's caused because of 3 bases being deleted out of a gene. Now, some alleles that cause disease may also have benefits, and the sickle cell gene is one of those. Carrying 1 copy of the sickle gene provides protection from malaria. Now, malaria is a disease that is carried by the Anopheles mosquito, and it's generally carried in very, very tropical, hot areas. So, guess where this gene predominates? It hangs around. It hangs around in very, very tropical, warm, moist areas. So that's why people in that particular part of the world are going to have more sickle cell anemia problems, because that gene is also protective of the people that are heterozygous. In other words, having a normal gene and then 1 sickle cell gene. Carrying 1 copy of the CF allele provides protection from malaria and actually from a couple of different things. So what we have is a deleterious gene that actually gives us some benefit along with the deleterious effects. Okay, so those are just some examples of abnormalities in human beings that come about because of chromosome abnormalities and because of genetic gene abnormalities. One of the things that we talked about in our last faculty meeting is where we wanted to end up. I only have a few more classes left with you guys, so what I would like to do, we've covered a lot of genetic material, okay, but in order to get through the material that we got through last year, we need to stop really where we are right now, and we need to move on to evolution. I'd like to spend the last few classes on evolution, so we're not going to have a test over this, and I know that disappoints everybody, but hopefully you've learned something here. Along with this lecture today, I'm going to send also a video that talks about reviews non-disjunction. So the next thing that we're going to be doing is we're going to be going through the next chapter on evolution, which would be 17, I believe. We'll be going through Charles Darwin, one of the last of the famous biologists that I told you need to remember. Charles Darwin is probably the most famous person in biology. Everybody recognizes that name and associates it with evolution. So we're going to finish up talking about what evolution is and how it impacts us as humans. So adios, amibas.